Please welcome Markus Schick, Technical University Munich, Charité Universitätsmedizin Berlin. Functional interpretation of omics studies.
We want to develop precise therapeutic strategies for B-cell lymphoma patients. And actually, we are in an amazing situation. Thousands of patients are fully characterized by means of whole exon, whole genome, or whole transcriptome sequencing. And for the coding genome, we have a pretty good idea of what's going on. However, the massive amount of genetic and, more importantly, non-genetic alterations which we find in tumor samples is facing us with a big challenge. How can we assess the quality of genetic alterations, and how can we predict if an alteration functionally contributes to tumor biology and ideally represents a therapeutic target structure or allows patient stratification? To tackle that challenge, we generate huge data sets of functional relevant alterations by in vivo screenings in mouse models of B-cell lymphomagenesis. We thereby activate and inactivate every single gene in the mouse genome and ask the same question: does this alteration cause cancer? We did this for aggressive B-cell lymphomas. Here we identified around 900 functional relevant alterations. More than half of them are also detected in humans. We then use the information from our functional data sets to interpret what we find in omics studies. For example, the 6q deletion, a long deletion affecting around 100 genes and thousands of patients every year. The huge amount of genes on this deletion makes it very hard to pinpoint the reasons for breaking of this chromosome. By combining our in vivo screening data and omics data, we could identify the gene SAM6 as one of the relevant targets of this deletion, and we could also provide genetic evidence that loss of SAM6 does indeed cause cancer. Beyond that, we also claim that we need to understand the mechanisms downstream of these relevant alterations. For SAM6, an enzyme with catalytic activity, we identified around 200 physical interactors. But frankly, this was not telling us much about tumor biology until we used the functional data from our in vivo data sets to inform our mechanistic studies. It then became very obvious by eye and by statistics that the cohesin complex is the tumor-relevant target of the 6q deletion, and we now rationally target this pathway. Taken together, we claim that we need to combine omics studies with functional data to then, on the one hand, identify biomarkers, and on the other hand, and maybe even more important, understand the mechanisms driving these tumor subtypes to then rationally develop treatment strategies. Thank you very much.